Welcome. My name is Scott Fountain. I'm Executive Director of North American Laboratory Sciences and Clinical Support at Charles River Laboratories. I would like to welcome you to our panel discussion today around analytics and bioanalysis and contemporary topics related to that theme. And with that, I would like to welcome our guests as they introduce themselves.
I'm Sam Islam from QPS. I'm the Senior VP and Global Head of Preclinical Development.
Hello, I'm Roger Hayes. I'm the Senior Vice President for DMPK at MPI Research.
Hello, my name is Yan Zhang from Bristol Myers Squibb. I'm Director at the Bioanalytical Science Department.
Hi, my name is Ola Saad. I am a Senior Scientist in Bioanalytical Sciences at Genentech.
Hello, my name is Afshin Safavi. I'm the co-founder and Global Chief Scientific Officer at Bioanalytics.
Well, I appreciate everybody's time today in wonderful Los Angeles, Hollywood, California. And I'd just like to jump right into the topics today. One of the things with bioanalytical laboratories, whether it's at the innovator site or CRO and even with the vendors, is the question of when do we bring in new technologies, sort of how close to the bleeding edge of implementation of new technologies do we get, and what's the return on investment. So I'd just like to open up with a conversation of when do we bring in new technologies, when's too soon, when's too late, and when is the sweet spot?
So as a CRO, the way we look at a technology, we don't bring a platform or technology for the sake of having a technology. There are a lot of good platforms out there, but for us, we have to make sure that the technology is mature enough and the instrumentation is robust enough that we can support many studies for various clients. That's one. And the second thing for us is there has to be a market for it. So usually it takes a few years before the pharmaceuticals adopt the technology to put some studies on it and eventually make it from preclinical to clinical, and later on they are looking to outsource obviously maybe the regulated studies. And that's when the CROs, at least for us, we get involved, we think about bringing it in. But at the end of it, it gotta be a reliable platform that we can generate quality data.
So if you're a vendor of hardware, everybody talks about disruptive technologies, and please invest in this great technology. But obviously in the CRO, it's a very different thing. It's more about the opportunity for actually using that equipment for generating revenue. And to have a sponsor request something that is matching what might be in their lab often determines when you're investing in that equipment. So there's obviously a lot of really good equipment out there, but as Afshin said already, it's really about having a platform that will give you a return on that investment, longevity, and certainly matches what the bulk of your sponsors are looking for.
Yeah, so from our perspective, I would say we have been closer to the bleeding edge, and sometimes it works out, sometimes it doesn't. I think the most important part for us is that it's okay to be close to the bleeding edge as long as we have the right people to run the equipment, and also making sure that we have the right regulatory component to address future opportunities. I think most of the time nowadays, a lot of the sponsors, a lot of the vendors, not like 20 years ago, and it says, hey, I have this box. Yeah. They sort of like tested the box out a few times. They beta tested, alpha tested. So I think now it's sort of less risky for us.
Yeah, just to add to that, I think the idea of total cost of ownership is important. So it's like, okay, new technology, and I liked, Zamus, how you touched on, well, is there a unique subject matter expertise I need to bring on board to actually be able to competently pursue this technology? What are the operating costs? Is it scalable? Is it scalable from a compliance perspective, from a business risk perspective, a scientific risk perspective? So I think those are excellent points as you think about the tool holistically.
And from our pharma industry point of view, we take a little bit more risk than CROs, and also depends on different stages. So if for this, because my organization, we have both discovery support and regulatory bioanalysis support. So we take more risk at the discovery bioanalytical support in looking into new technologies. We we look at the technology in two ways. One is to see whether technology can address our bioanalytical gap. So and then another thing is we look at the technology to see is this bring some idea or some new way of approaching our problem which we haven't thought about that. So at the discovery we didn't think too much about compliance issue, but mainly on more technical and scientific issue. Then we evaluate at the discovery stage. But when we decide that one, we will put it into more production stage. So we evaluate more on the long-term value and also whether it can be implemented in the CRO.
So do you have a mandate from your management to actually bring in new technologies?
Yes, we have in our department, we have a small team to look at technology evaluation. So we have idea, now we discuss it in our team, and we have different criteria looking into the technology. For example, whether it's address our issue or whether bring new sort of thinking or new approach or improve our productivity, cut costs. So we have different criteria. Now we evaluate that and also bring technology to test that, to show, and we use some case study to test that technology. Then we will decide whether we are going to purchase.
So also speaking from a biopharmaceutical perspective, we're actually encouraged very similarly to be very proactive. So to go out there, our scientists, our researchers go out to conferences, go out to these meetings to actually look for all of these opportunities and new technologies that are on the bleeding cutting edge, and to bring them in and to share with those at the company how they think those could contribute to improving whether it's sensitivity, resolution of the work that we're doing, throughput or whatnot. And so, yes, there's actually dedicated time and emphasis that's put on that. And in terms of our collaboration then with CROs, we actually do, look for those CROs that are taking that risk to be a little bit upfront and trying to, when we're ready to collaborate on something in the more development stage, to be able to work with them closely. But to Jan's point, we have really open wide range in the discovery range. There's a lot of interesting scientific questions, a lot of opportunities to do cutting edge work, publish that work on programs that we're able to, and we're really encouraged to do that. So we are always looking actively at cutting edge technologies.
For us as a CRO, I can tell you that as scientists, we love to play with new toys. There is no doubt, right? We all like to do that. For us, we also have to balance that as exactly when to invest in that technology, right? If you invest too early during, let's say, even a beta testing, we can get burned and really sometimes end up with instrumentation that is still not mature for the market. On the other hand, if we enter into the market, and I think to your point, that's a good point, right? You want to be on a cutting edge, but I think if you go in there too early, it can hurt you. But if you go too late, you can be behind. So it's a balancing act, and the CROs that have been successful, I think, are the ones that work closely with their pharma partners to know when to enter in that market. And I think you brought up a good point around that. Once you start putting this instrumentation in the regulated environment, for us, we have basically accepted that. Whatever the cost of the instrument is and the amount of time it takes to implement to actually the hardware part, probably it takes 2 or 3 times more funding and time to do the software validation to 20 Part 11 and the regulatory requirements and incorporate that with your LIMS system. So I think those are also considerations that we deal with every day.
Yeah, so it's an interesting perspective when you look at vendors putting in demo equipment or demo instruments. Would you please evaluate this? And to see where are they putting that equipment in? Are they putting it in at CROs for beta testing or for perhaps alpha testing, as Amos was talking about? Or are they putting it into pharma because that's perhaps where they might actually get that sale when that instrument maybe works out? So I'm just curious to see, how much we're all putting in or doing that beta testing for vendors, or are they going to pharma first?
So we we never have a quote unquote a beta instrument.
Right.
We have instrument that has been out there, but whether they have been applied to the same type of methodology, the same type of
Right.
things we need to address is different. We are actively beta testing new software
Yeah.
because there's a lot of current software that doesn't address fundamental feature that we need to address, right? So there is all these lovely software that you can do quantitation of small molecule, but quantitation large molecule, the software is not quite
Right.
ready yet. Especially start looking at intact, you're looking at metabolism of these biologics, it's very different. So we've been very active in that area, and software is not cheap, right? And then the other thing is, addressing the fact that you're acquiring the data and then packing it in a computer somewhere and then download it into some type of compliance component would like to be addressed at right front,
Right.
because it impacts IT way too much.
Sure. Yep.
I believe one reason that vendors do sometimes choose to work with biopharma in these cases is a lot of times we will be working closely with them, bringing them actually some of our pain points and saying, you know, here's a problem. I wish I had software that could do X, Y, or Z, as you were indicating, Zemus. So I'd like to be able to have software that could help me interrogate these large molecule biotransformations, and this is what it would, you know, in my world, be able to do for me. What do you think? Is there something in your book? And we've engaged with vendors that way, whether it's, you know, or I need better resolution at this mass range, you know, what can be done here? And so that's in the instances that we've actually had, you know, beta testing done in our labs and instruments or software that's been brought in, it's been brought in based on here's a need that we express that we'd like to try to see met in some way. And those vendors that are willing to sort of step up and provide us with something that we can try out, we're really excited to do that.
Yeah, it's similar in our company. We partnership with a lot of vendors to not only beta testing, but also further optimize the technology. Sometimes we identify the issues that then they come have their scientists come to work with us to develop an application, and for them it's advantage. So they get more application development, and for us, and we can utilize their expertise to solve our issues. So I don't know which one they reach first, but it definitely in pharma we work with them in many cases.
Yeah, so kind of the question is who's driving innovation? Is it pharma or the CRO?
Yeah, and I would say it's a partnership, and I've seen real nice examples where you can actually have a trinity, right? I mean, the vendor and the CRO and the innovator collaborating around solving a common project.
Right.
So, so that we talked a lot about then sort of the hardware and that capital, and there's also the human capital. So maybe either with analytics, either at the innovator side or the CRO side, if people have thoughts or perspectives around, you know, identifying talent and then recruiting, retaining, training. I know there's a lot of growth in analytics right now and the needs in the industry. I'm sure it's something that we're all struggling with, but does anyone want to add or share a thought around identifying and retaining top quality talent? Because we're all competing with each other, frankly, around our talent.
I think we all are in agreement that the universities are not necessarily putting out exactly the people that will do well in the disciplines that we work in, right? Nobody comes out knowing how to develop and validate a PK or immunogenicity or biomarker or NAB assay, right? So, and there's a limited number of people out there with that level of experience and expertise. So I can tell you the approach we have taken. We have actually, you know, again, just a traditional recruiting and all that goes on. But the key is, once you identify those individuals that you find the talent in them, bring them in, have a training program, beyond just reading a couple weeks of SOPs. So we have actually initiated what we call Bioanalytics University. It is about 10 or 12 modules. It's about 1 to 1.5 hour long. And we take them through every step. And it's once a month or sometimes a couple times a month. And so we are in essence providing continuing education internally within the company. And to us, we finally started doing this and it is very effective. So to me, that's one. Now in terms of retention, if you look at, so today I'm happy to tell you that we have over 90% employee retention. It's one of the highest in the market. Wow. The way we do that is not because we can compete, to be very frank with you, with the compensation that some of the pharmas might be able to provide. It's about, and it's not that the employees are not working very hard. It is an environment that is a family-oriented environment, that people coming, majority of the joy they get is working with different clients, different projects, and within the company, having that camaraderie, the level of trust that I watch for you. And so it is great that I can tell you probably about over 95% plus of my senior staff and scientists that started with me, you know, almost a decade ago, they're still with us. So it takes a lot of time and effort, but I'm very curious to hear about, you know, from the other folks and learn from you guys.
Do you bring them in? What's their first role? So you bring them in and you say, okay, you're a method developer, or you bring them in, put them into production.
So what we do to, maybe it's a little excessive, but we actually, during the interview process, we sort of try to see what is it they're good at. And by that, we actually give every employee, doesn't matter, bachelor, master, or PhD, 0 or 20 years experience, they have to take a written test. We got to understand what are the gaps. You can have PhDs that still do not know how to do a simple conversion. And it's okay. And in some instances, we have bachelor level coming out of school that they are amazing. And then we also give them a hand pipette test with food coloring. We try to assess them, right? And also we ask them, what is it that they like to do? Taking all that into consideration, we also got to see what is the type of demand that is coming from the market, and try to balance that. I don't think there is one solution that fits for every pharma, biotech, or a CRO. This has worked well for us, and we're just trying to see how can we keep this culture and this system as we grow, right? The key is, as you grow, maintain what is working. So,
yeah, I think we take kind of the same approach where you try to do your best, but a day interview is how much can you really achieve, right? So it's that ask the right questions, but then you'll get the younger generation that will come in, and their patience for career development is much shorter than you or I. Where 6 months in the laboratory, it's like, well, okay, I've learned everything there is to learn about this assay or this technique, so now I'm ready for management. And that retention is a challenge, because now we're doing routine analysis is thought of as not a good thing, right? And yet there is a generation that were doing routine for 10 years. And we're very comfortable doing it. So for us, it's that challenge of, well, we need to do routine. This is what we are doing as a CRO. There is production work. But then to make it either through cross-training or just introducing new techniques or something different completely, go from large molecule to small molecule or something like that, to maintain their own sense that I'm doing something that's not routine. Because what we found is that when you do routine, they don't have a lot of patience, and so 6 months to maybe a year is when they start chafing and are looking to move on.
Yeah, so from very similar experience, we also close to universities, so finding university students for the first entry level is relatively easy. SOPs is pretty boring to read. Consistency between people to people is difficult. So we essentially put them through a training team. We actually have a training team for each department. And the group of the trainers are consistent to make sure that each person coming in will be able to function specifically. And they're given about 6 weeks generally. So if they're really fast, they're 4. If they're really slow, is 8, but average is about 6 weeks. As Roger says, after a while, people do get bored, right? I mean, people do get bored. So in a way, we're lucky, although our group, our 3 big group is relatively the same size, and the people from small molecule are allowed to go to DMPK. And the DMPK guys do go to our ELISA group. Our ELISA group can also go to our small molecule group. So from that perspective, there is some transitioning over. People that wants to do it and have ability to do it are allowed to do it.
Right.
But again, it's sort of, sometimes people just are comfortable, other people are not so comfortable, right?
Right. Well, there may be retention within the company. I think that's the secret, is that you get retained within the company, but not necessarily in the role that they were originally potentially recruited for.
Yeah, I absolutely agree with that. For in our company, our department, we have quite good luck to retain people, and also in comparison with the CRO, we have more diverse opportunities. So that allow people not only the new one to start with one role. We have cross-training opportunities. Really depends on the motivation and also the passion, and also what that person good at that. And some people actually, they love just to do routine, and then they happy to live there, so we have opportunity just to do routine production work. And some people more like interact with the team, so we have a role to interact with the team, and if demonstrate good about that. And we also have a different technology platform, so LCMS and liquid binding assay, and then we have different kind of bioanalytical opportunity, from discovery support to regulatory support and immunogenicity biomarkers. So this kind of, I think that's we are in the very good situation, and also to allow people to develop, and also the company as a culture. I think BMS is very good at developing people. We have people strategy. Every manager in their objective has to demonstrate how you play the role in developing your people. I think that's also very important. So we have it's a part of our objective to be evaluated at that. So it's big on people development. So that's the one part, and we doing quite successful to repair.
Yeah, I would have to agree. I think at Genentech that is something that's taken quite seriously. Genentech is and has been ranked as one of the great workplaces and best places to work for many years, as you might know, based on Science and Fortune magazines. And so there's a strong emphasis on that. I think a lot of people at Genentech really value the science-driven culture, the patient focus, and that there's never a chance that our management doesn't choose or use an opportunity to reemphasize that, whether it's patient guest speakers that come, or at every level of the organization, making sure that people know what they're doing and how it contributes to the end products that we're developing. I think in terms of looking at the talent that we are recruiting, you know, a lot of times we look for very specific talent. Maybe we're looking for someone who has protein mass spec experience or something. And so, you know, things like that are growing out there as more and more people start doing more cutting edge work. And so sometimes it's actually getting a little bit easier to find people with some of the skill sets that we're looking for, whereas in the past it's been a little bit more difficult when there's fewer people out there who have that experience. But once they're there, as Jan mentioned, at Genentech we have many opportunities. There's diversity in terms of the type of work that's being done, whether it's using different technologies, using mass spec, using ligand binding assays, diversifying the assay types that you're developing, maybe switching from PK to ADA assays, or PK biomarker as well. And so there's this diversity, also the different types of roles that bioanalytical roles that can be played. So whether it's helping with the outsourcing or whether it's working with another team, there's job rotation opportunities, so they can take short rotations in another group and learn what that group does as well. One thing I also would emphasize is our bioanalytical folk also serve as actual integral members of the drug development teams. So we have bioanalytical working groups that meet regularly to develop an actual bioanalytical strategy that is meant to support any drug in our pipeline. And we even have bioanalytical scientists who also will lead these cross-functional teams as well as our pharmacology sub-teams. So they will serve not just as a set of hands generating necessarily just data that we hand over the fence, but we will actually be participating integrally in that bioanalytical strategy, explaining it to the team, and perhaps even leading a cross-functional team.
You know, Scott, part of the question that I think is interesting, you say, you know, around retention, right? And you know, I think we can set up the career ladder and a lot of other things that we talked about. What is interesting, at least for me, what we're finding to be effective may not work for every company or their size. For example, we basically started 3 years ago having fresh foods for all the employees every day. That's one of the things that they wanted. We started, you know, having small rewards that, for example, if you hit a milestone, one of the best things that employees like, that we hired a company to come and wash and detail every employee's cars, right? So if you have a mom that, you know, has, or a dad that takes the kids to school every day and the car is a mess, you know what happened, you know, just getting the car clean is what they appreciate. The cost to the company is very small, right? Some of the other perks, for example, we found out and we actually wrote down, where do the employees like to eat? Where do they like to shop? And we actually bought gift certificate that, you know, we hand to them. And it doesn't have to be much, $50 or $100. But the recognition that, good job, you did a great job, the client is happy. And it's interesting, the most important thing is you gotta find out if some employees, they like public recognition. Right. But as scientists, unlike some of our BDs, right? We sometimes like private recognition. Right. To have your boss take you to the room and say, thank you, shake your hand, and take the family on a dinner. So I think, you know, sometimes we think about, you know, opportunities or career ladders or things that may cost a lot of time and money, but those little things mean a lot to the employees.
Right. Yeah, those non-material congratulations and attaboys and things like that.
Yeah.
Well, thanks. I appreciate everybody's time and discussing instrument capital and human capital, and I was really encouraged to hear how people are thinking about diversifying both our skills and our partnerships so that bioanalysts feel like they're 100% part of the team, and also having the opportunity to lead teams, not just producing data per se, but also knowledge and integrating knowledge. Well, thanks again very much, and we appreciate your time, and I appreciate everyone's contributions. Thank you.
I know we're filming, it's not the time around.
Let's do it, team. Whoa! Woo!
Tell you, like, stay.
That's all right.